What is cognitive neuroscience? Cognitive neuroscience is the integration of brain and behavior. Throughout this semester, we will discuss the neural mechanisms underlying behaviors that mediate emotion, language, memory, and sensation and perception, among others. Before we get to these processes, we will dive into the semester by covering a thorough overview of nervous system anatomy, and particularly the regulation of neuronal activity. It is so imperative to gain a clear understanding of neuronal activity and how brain regions communicate and coordinate together in order to have a deeper understanding of cognitive control. Knowing the structural framework that supports behaviors allows for scientists to manipulate those structures in order to gain therapeutic effects. We will discuss what instruments are used in order to study neuronal processes in the context of cognitive neuroscience. To begin this week, we will talk about the historically relevant scientists and physicians who have made a major impact on the field of cognitive neuroscience in order to understand how these developments have allowed for the advancements in the field today. The integration of brain and behavior, that's what we mean by the term cognitive neuroscience. This is a field devoted to studying how the brain works, how brain structure and function affect behavior, and ultimately how the brain enables the mind. Although the study of brain and behavior interactions started as far back as the 1600s, it was not until the 1970s that the field was given its name. And in fact, Gazzaniga himself, one of the authors of this book, was involved in coining the term for the field. This occurred during a taxi ride to dinner with the cognitive psychologist George Miller. Gazzaniga and Miller came up with the term cognitive neuroscience from the words cognition, or the process of knowing, and neuroscience, or the study of how the nervous system is organized and functions. This is the perfect term for the field, and it took hold and has persisted within the scientific community. When considering how the brain functions, it is important to think about the process of evolution. While human brains have been around for about 100,000 years, the primate brain appeared between 23 to 34 million years ago. Now, the human lineage diverged from the last common ancestor with the chimpanzee around 5 to 7 million years ago. And ever since, our brains have evolved into the present human brain, adapting along with the environmental pressures throughout the years. As we go through this semester, I want you to keep the following questions in mind. Why might certain behaviors have evolved? And what's the benefit to humans for that evolution? How might those changes promote survival and reproduction? As humans evolved from the hunter-gatherers to a life where day-to-day activities did not revolve around survival, people started to think more about causation and the construction of complex theories about the motives of human behavior. The dominant theme of cognitive neuroscience remains: does the brain work as a whole or as separate parts working independently? Scientists like Thomas Willis shed light on this with the notion that isolated brain damage could affect behavior. Willis contributed greatly through his anatomical drawings of the human brain as depicted on the right. Another century later, Franz Joseph Gall showed that the brain was the organ of the mind and that innate faculties were localized in specific regions of the cerebral cortex. He also found that greater frequency of specific activity meant growth of brain area responsible for that activity. Physiologist Marie-Jean-Pierre Flourens found through animal studies that when he removed cerebral hemispheres, the animal no longer had perception, motor ability, or judgment abilities. Also, without the cerebellum region, the animals became uncoordinated and lost their equilibrium. He developed the aggregate field theory, such that the whole brain participated in behavior. In 1836, a neurologist named Mark Dax performed clinical studies that demonstrated speech disturbances in patients with left hemisphere lesions, identifying a left hemisphere specific role for speech production and comprehension. Meanwhile, in England, the neurologist John Hewlings Jackson began to publish his work on behavior of persons with brain damage. He found that during the start of seizures, some epileptic patients moved in such characteristic ways that the seizure appeared to be stimulating a set map of the body in the brain. The contractions in muscles caused by uncontrollable firing of neurons progress in an orderly pattern from one body part to another, suggesting the role of a body map or topographic organization of the cerebral cortex. This means that each specific area of the cerebral cortex, later known as the motor cortex, is important for controlling a specific area of muscle contraction. We will revisit this in later chapters. It was also during this time that Parisian physician Paul Broca and German neurologist Karl Wernicke were studying patients with speech problems. Broca worked with an individual referred to as Tan because this man had developed aphasia, where he could understand language, but the only word that he could utter was Tan, leading to studies of a brain area where Tan had a lesion, now called Broca's area. Wernicke reported on a stroke victim who could talk quite freely, but made little sense when he spoke. He had a lesion in a more posterior portion of the left hemisphere, now referred to as Wernicke's area, which is largely responsible for speech comprehension. These studies supported the notion that focal brain damage causes specific behavioral deficits. Broca's area demonstrated here, whereas Wernicke's area is back here. German neuroscientists began to study the brain using microscopic techniques, which enabled a close view into the tissue and cellular architecture. Jan Evangelista Purkinje described the first nerve cell in the nervous system in 1837. In the 1900s, cellular architecture was further explored with the aid of particular stains like the ones developed by Nissl and Golgi to allow for viewing of a cell under a microscope. Cajal identified neurons as discrete entities and found that electrical transmission occurred in one direction. This opened up discovery of neuronal networks and how neurons communicate across a synapse. From here, it was evident that knowledge of the parts must be understood in conjunction with the whole. One must study all parts of a mechanism in order to understand how they work together. This is a theme that will be carried out through the semester. Though certain brain regions are important for specific tasks, it is the function of the brain as a whole, or networks of cells within those regions communicating with others, that allows for full brain control of behavior. Before the start of experimental psychology, Many philosophers studied the nature of knowledge and how we came to know things. Two main positions were held, that of rationalism versus empiricism. Rationalism meant that all knowledge could be gained through the use of reason alone. They believed that truth was intellectual, not from a sensory experience. Whereas empiricism means that all knowledge comes from sensory experience. The brain is essentially a blank slate where direct sensory experience produces simple ideas and concepts and shapes that brain throughout the years. An idea that stems from empiricism is associationism, or that a collection of a person's experiences determines the course of mental development. John B. Watson was important in this field and came up with the popular idea that learning was the key. He noted that everyone had the same neural equipment on which learning could build. Psychology could be objective only if it were based on observable behavior. American psychologists focus on behaviorism. However, Britain and Canada psychologists were focused on electrophysiological study. Herbert Jasper and Wilder Penfield used the Montreal procedure in treating epilepsy, in which they surgically destroyed neurons in the brain that produced seizures. Donald Hebb in Nova Scotia demonstrated that neurons can combine together into a single processing unit, and the connection patterns of these units make up the brain's response to a stimulus. Cells that fire together, wire together. The 1950s marked the end of behaviorism and stimulus response psychology, as psychologists began to think more in terms of cognition, not just behavior, based on these studies. Thus began the field of cognitive neuroscience as a distinct multidisciplinary field. Scientists used a variety of approaches, including biochemistry, anatomy, electrophysiology, pharmacology, and behavior, in order to study cognition and how the brain and behavior interact. For example, Patricia Goldman-Rakic in the 1970s performed multidisciplinary studies using a wide variety of techniques to demonstrate the role of the prefrontal cortex area in working memory as well as in dopamine neurocircuitry. Her studies caused a major shift in the understanding of mental illnesses, especially schizophrenia, which was thought to be due to bad parenting. In 1875, a galvanometer was used to measure continuous spontaneous electrical activity from the cerebral cortex and skull surface of live dogs and apes. A string galvanometer was able to make photographic recordings of the electrical activity. And in 1929, a German psychiatrist, Hans Berger, published recordings of human brain electrical activity and named the recording an electroencephalogram. This remained the sole technique for non-invasive brain study for a number of years. Scientists discovered that blood flow followed function. Thus, a way to understand brain activity was to measure blood flow in a particular area. Methods were used to develop positron emission tomography, which uses radio tracers rather than inert gas. Computerized axial tomography used mathematical techniques and an X-ray scanner to develop 3D images of the brain from a series of transverse images. Positron emission tomography used radio tracers for metabolically active molecules in the brain, like oxygen and glucose, in order to measure blood flow and metabolism in specific areas of the brain. Magnetic resonance imaging is based on the principle of nuclear magnetic resonance. Researchers demonstrated that following injection of contrast material into the bloodstream, changes in the blood volume of a human brain produced by physiological manipulation of blood flow could be measured using MRI. This technique produced excellent anatomical images of the brain. Functional magnetic resonance imaging allowed for functional mapping of the brain by essentially combining PET scanning techniques with MRI. Again, this technique relies on the fact that cerebral blood flow and neuronal activation are coupled. In conclusion, this chapter offers an introduction to the big questions and discussions that we will be going over throughout the rest of the semester. We'll next continue with an overview of brain anatomy and cellular mechanisms. We'll discuss more of the methods available for use in observing mind-brain relationships. We'll discuss core processes of cognition, like learning, language, emotion, etc. We'll also talk about cognitive control, social cognition, and free will. And you'll notice that each chapter begins with a story, followed by discussion and further reading for those who are interested. So please take advantage of the chapter readings, as well as some of the additional readings that are posted at the end of the chapter. I want you to keep in mind a couple of questions from this chapter material only. Can we study how the mind works without studying the brain? Also, how do you think the brain might be studied in the future, given the trajectory of brain study over the course of the past, hundreds of years?